Unraveling the Role of Epigenetics in Environmental Genomics. Title: Unraveling the Role of Epigenetics in Environmental Genomics. Within the realm of environmental genomics, epigenetics plays a pivotal role by investigating how external factors influence gene expression without altering the DNA sequence itself. Understanding Epigenetics, 200 words. Epigenetics encompasses a range of molecular modifications and mechanisms, such as DNA methylation, histone modifications, and non-coding RNA molecules. These modifications can affect gene activity and have a lasting impact on various biological phenomena, including development, aging, and disease susceptibility. Unlike genetic mutations, epigenetic alterations are malleable and can be influenced by both genetic and environmental factors. Environmental Genomics and Epigenetics, 300 words. Environmental genomics aims to decipher how genomic responses to environmental stimuli contribute to an organism's phenotype. Epigenetics provides valuable insights into the mechanisms by which the environment can influence gene expression patterns. Exposure to various environmental factors, such as diet, toxins, stress, and pollutants, can lead to changes in DNA methylation patterns or histone modifications, altering gene expression regulation. Role of DNA methylation, 350 words. DNA methylation, the addition of a methyl group to the DNA molecule, is a crucial epigenetic mechanism in environmental genomics. By altering the accessibility of genetic information, DNA methylation can modulate gene expression patterns. Epigenetic changes through DNA methylation have been linked to various conditions, including cancer, cardiovascular diseases, and neurological disorders. These modifications can result in chromatin remodeling, impacting gene accessibility. For instance, exposure to environmental stressors has been shown to induce changes in histone modifications, leading to changes in gene expression patterns. Current challenges and future directions, 200 words. The field of epigenetics within environmental genomics is evolving rapidly, but it still faces numerous challenges. The interpretation of epigenetic modifications in the context of environmental exposures remains complex, and the identification of causative relationships is challenging. Certain environmental factors can alter gene expression patterns through epigenetic modifications, increasing the risk of developing various diseases. Understanding these epigenetic changes can potentially pave the way for targeted interventions and personalized medicine approaches. Through genome-wide studies, researchers have been able to identify specific loci where genetic variation modulates the response to environmental factors, leading to differential gene expression. Epigenetics and environmental health, 200 words. The field of environmental health has also benefited from the integration of epigenetics. Understanding how environmental exposures can modify the epigenome has the potential to inform public health strategies and policies. Additionally, epigenetic biomarkers have emerged as potential tools for assessing exposure history and predicting individual susceptibility to environmental factors. Technological advances in epigenetics, 250 words. Continued advancements in technology have significantly contributed to the progress of epigenetics and environmental genomics. Epigenetics and therapeutic targets, 300 words. The insights gained from epigenetic research and environmental genomics have opened up new possibilities for therapeutic interventions. Epigenetic modifications are reversible, making them attractive. Targets for drug development. Researchers are actively exploring the use of small molecules or gene editing technologies to restore or modify epigenetic marks associated with disease or environmental exposures. By precisely targeting these marks, it may be possible to restore normal gene expression patterns and potentially reverse the adverse effects of environmental exposures. The role of epigenetics in developmental programming, 300 words. Another important aspect of epigenetics in environmental genomics is its role in developmental programming. Epigenetic modifications occurring during early development can have long-lasting effects on an individual's health and susceptibility to diseases later in life. Similarly, exposure to maternal stress or other environmental factors can induce epigenetic changes that impact the offspring's neurodevelopment, increasing the risk of mental health disorders. Understanding the underlying mechanisms of developmental programming through epigenetics is crucial for mitigating the long-term effects of adverse environmental conditions. By identifying specific epigenetic modifications associated with developmental programming, researchers can develop targeted interventions to promote healthier outcomes. Additionally, this knowledge can inform public health policies aimed at improving maternal and child health. Traditional risk assessment methods often focus on direct interactions between environmental agents and DNA sequence variations. However, epigenetic modifications can occur without changes in the underlying DNA sequence, yet still have significant implications for gene expression and disease susceptibility. By incorporating epigenetic data into risk assessment frameworks, it is possible to better estimate the effects of environmental exposures on human health. For example, monitoring DNA methylation patterns associated with exposure to certain pollutants can serve as early biomarkers indicative of potential health risks. This can enable prompt actions to reduce exposure or implement preventive measures. Epigenetics also holds promise in predicting individual susceptibility to environmental factors. Unlocking the potential of epigenetics in precision medicine, 300 words. The incorporation of epigenetics into precision medicine is a growing area of research with immense potential. Precision medicine aims to tailor healthcare decisions and therapies to individual patients based on their unique genetic makeup and environmental exposures. Epigenetics offers a deeper understanding of how environmental factors can impact gene expression and disease susceptibility, providing critical insights for personalized treatment strategies. By analyzing a patient's epigenetic profile, it becomes possible to identify specific epigenetic marks associated with disease susceptibility or response to certain treatments. This information can be used to guide therapeutic decisions, ensuring that patients receive treatments that are most likely to be effective for their specific condition. Additionally, epigenetic biomarkers can serve as indicators of treatment response, allowing for real-time monitoring and adjustment of therapeutic interventions. Furthermore, epigenetics can shed light on the underlying mechanisms of drug resistance. Epigenetic changes can influence the expression of drug targets or drug metabolism enzymes, potentially rendering certain treatments ineffective. Understanding these epigenetic modifications can help identify the molecular mechanisms driving drug resistance and guide the development of targeted therapies to overcome resistance. Epigenetics and nutrigenomics, 250 words. The field of nutrigenomics explores how dietary components interact with an individual's genetic makeup to influence health outcomes. Epigenetics plays a crucial role in nutrigenomics by elucidating the mechanisms through which nutrients can modulate gene expression patterns. Various dietary factors, such as vitamins, minerals, and bioactive compounds, have been shown to impact the epigenome. For example, folate, a B vitamin found in leafy greens and legumes, is an essential component in the 1-carbon metabolism pathway, which influences DNA methylation. The study of nutrigenomics and epigenetics has revealed the potential for personalized nutrition interventions. This personalized approach to nutrition can guide individualized dietary interventions.